Hi, I'm Tim Studd. I'm the editor-in-chief of Laboratory Equipment Magazine, and we are at LabAutomation 2010, and we're here in the Biolog booth with John Pietrzyk, who's going to tell us about some products that he's exhibiting at this booth. John.
Thank you very much. It's a pleasure being here. Yes, thank you for this opportunity. Biolog is a company based in Hayward, California that specializes in providing cell-based assay systems for metabolic profiling. The company has been around for 26 years. It's been profitable for the past 24 years. It has 55 employees. And historically, it has emphasized the microbial identification using metabolic profiling. And we are here today presenting an extension of that technology platform where we're looking at transitioning over into the new space, the mammalian cell biology space. But in essence, the technology platform that we're using works in both spheres. We use 96 well plates, and in each of the plates, each well has a unique energy substrate. So on the panel behind me, the top 4 96 well plates that you see, each of those wells has a different energy substrate. In fact, among the 4 plates, we have 367 different carbon and nitrogen energy substrates. Each one is indexed to a unique metabolic pathway. And we use a redox reaction that measures NADH generation on a pathway-by-pathway basis. So in the course of 1 to 4 hours of incubation with cells and with the redox dye added to each of the wells, we are able to generate a unique metabolic fingerprint as displayed on the panel behind us. The technology is being used, historically I should say, it's been used for distinguishing very closely related microbial species. It turns out that even very closely related species show a selectivity in the substrates that they are able to metabolize. And the same seems to be the case with mammalian cells. There are 300 unique mammalian cell types in the human body. And to date, in our hands, we've been able to demonstrate that these cells show selectivity in which metabolic substrates they bring on board and metabolize. The technology is used in optimizing media formulations, in identifying which clones or which cell lines may be best to work with in product development. We also have plates that are dedicated to toxicology assessments, looking at impacts to mitochondria, or to the glycolytic pathways. So it's an exploratory platform. You can explore different substrates to see how your substrates may affect metabolic behavior, or this is a plug-and-play solution whereby, for example, if you're doing QA/QC studies, and you want to know if the cell line that is in your hands is indeed the cell line that you think it is, if you have a metabolic fingerprint of it, before and after an operation, you can be sure that if the fingerprints match up, that you're dealing with the cell that you think you are. So it has an important cell identification capability, QA/QC in bioprocessing, and as I mentioned a moment ago, it has strong applications in formulating media which optimize the metabolic behavior of cells. So again, it's a pleasure being here. Thank you so much for allowing me to present our technology.